Somewhere beneath the Arctic surface, an animal can spend centuries moving through water that never sees the sun. The water is barely above freezing. The pressure is immense. There is almost no light, and almost nothing moves quickly enough to matter. Yet something has been passing through this darkness for hundreds of years. It can grow more than 7 meters, about 24 feet, and weigh more than a metric ton, roughly 2,200 pounds. It moves at less than 1 mile per hour through a world where almost nothing can follow it, and nothing in the ocean hunts it. This is the Greenland shark, the longest lived vertebrate ever confirmed on Earth. One individual was estimated to be around 392 years old, with an upper estimate reaching roughly 512. It may have been born before the Pilgrims crossed the Atlantic. For centuries, these sharks lived and died beyond human observation. Then, in March 2022, one appeared on a beach near Newlyn Harbor in Cornwall. A dog walker found her in the surf. She was nearly 4 meters, about 13 feet, long and weighed more than 270 kilograms, roughly 600 pounds. She was still alive. Her fins were torn. The soft tissue around her head was battered and bleeding, and she had swallowed sand before she died. But there was something even stranger about her. She was only about 100 years old. By Greenland shark standards, that was still remarkably young. She had not reached sexual maturity and had never reproduced. Then scientists examined her body and found something inside her skull that had never been documented in this species before. That discovery turned an unusual beaching into something much stranger. No one had ever been able to study the death of a Greenland shark like this. Not one that had spent a century in darkness, not one from a species capable of surviving for centuries, and not one whose final moments could reveal what had happened inside its brain. There was another problem. Her body contained trimethylamine oxide, a compound that makes Greenland shark flesh toxic when eaten fresh. This was an animal built for a world humans almost never enter: cold water, near total darkness, extreme pressure, and a heartbeat that can slow to only a few beats per minute. A life measured not in decades, but centuries. And then something went wrong. She left the deep. She entered a world she had almost certainly never experienced before, and she died on a beach. The question is not simply what killed her, it is what happened inside an animal that had evolved to survive for centuries in one of the most hostile environments on Earth. In this journey, we're following the final days of a Greenland shark to uncover what brought her out of the deep, what scientists found inside her skull, and what her death may reveal about the hidden pressures facing one of the ocean's strangest survivors. The Greenland shark has a shape that seems almost unfinished, as though the ocean had slowly worn it down until only the essentials remained. Its scientific name, Sumniosus microcephalus, means small-headed sleeper, a fitting name for an animal whose life is built around low energy, cold water, and long stretches of time. It belongs to the sleeper shark family, a group whose members are adapted to deep, dim environments where speed is rarely useful. Its world stretches across the Arctic and North Atlantic oceans. Greenland sharks are found from the Canadian Arctic and Greenland eastward towards Svalbard, Iceland, and the Barents Sea, following the coldest water available through a broad and shifting range. Their presence has also been recorded much farther south, including the Gulf of Mexico, where individuals can occupy unusually deep, cold layers far below the warmer surface. Depth is one of the defining features of this existence. Greenland sharks have been recorded from roughly 183 meters, about 600 feet beneath the surface, to approximately 2,647 meters, about 8,684 feet. They are not simply residents of one fixed level. Their position can shift with the seasons, moving deeper during warmer periods and rising somewhat when colder water becomes more accessible. The temperature around them can approach -2 degrees Celsius, about 28 degrees Fahrenheit, remarkably close to the freezing point of seawater. In this environment, nearly every movement carries the character of conservation. The shark does not need to hurry. Its body is designed to function while spending as little energy as possible. A large adult can reach about 7 meters, about 23 feet, in length and weigh more than 1,180 kilograms, about 2,600 pounds. That makes it one of the largest predatory sharks alive, yet its proportions do not suggest pursuit. The body is broad and heavy, with a mottled gray-brown surface that blends naturally into the darkness and muted colors of deep water. Cruising speed can remain below 1.6 kilometers per hour, about 1 mile per hour. For an animal of this size, that is extraordinarily slow. Yet there is little evolutionary reason for a healthy adult to become a fast swimmer. Mature Greenland sharks have no known natural predators capable of routinely hunting them. Their survival has instead been shaped around endurance, efficiency, and the ability to remain in an environment where few other large predators can comfortably persist. The lineage itself is ancient. Sleeper sharks have been present in the oceans for tens of millions of years, passing through immense changes in climate, ice, currents, and marine ecosystems. The Greenland shark is therefore not simply a large fish living in cold water. It is the surviving expression of an evolutionary strategy refined across geological time, a body tuned less for bursts of movement than for remaining functional when the surrounding world offers almost nothing for free. Its appearance reflects the same economy. The snout is short, the eyes small, and the fins modest compared with sharks built for pursuit. Nothing about the animal suggests acceleration. Its body seems suited to drifting through enormous distances while spending little energy. Even the water becomes part of its biology. At these depths, pressure rises dramatically, temperatures remain close to freezing, and sunlight disappears entirely. The shark occupies a realm where scent, vibration, and electrical signals can carry information through darkness, while slow movement conserves energy. There is also flexibility in where it can live. A species associated with the Arctic can appear far outside the polar region when the right combination of depth and temperature exists. The surface map gives an incomplete picture. Much of that home lies beneath ordinary ocean boundaries, extending into layers people cannot reach. For most animals, such a habitat would be difficult to endure. For the Greenland shark, it is simply home. The first clue is not in the shark's size, but in the rhythm of its heart. At rest, it beats only about 5 times each minute, a pace so restrained that the entire circulation seems to operate on a different clock from our own. A human heart may beat 60 to 100 times in a minute. The Greenland shark spends almost nothing on unnecessary motion, allowing each contraction to carry its blood through a body designed for extreme efficiency. That economy extends into growth itself. Its tissues accumulate new material at less than 1 centimeter, about 4/10 of an inch, each year. A young female can therefore spend roughly a century reaching 13 feet, and another several decades passing through the stages that eventually lead toward reproduction. Time is not merely something this shark experiences. Time is built into the rate at which its body changes. Inside that slow metabolism lies another remarkable feature. Measurements made through respirometry in the Canadian Arctic have shown an exceptionally low resting metabolic rate for a shark. The animal consumes very little oxygen when it is not actively swimming or feeding, Every cell is operating with the same principle as the whole body, spending only what is necessary and preserving the remainder. Cold helps make this possible. Water near freezing slows the chemical reactions that sustain metabolism, reducing the pace at which molecules interact and limiting the accumulation of some forms of cellular damage. Yet cold cannot explain the extraordinary lifespan by itself. Northern wolffish inhabit similarly frigid Arctic waters and can live for only a few decades. Deepwater six-gill sharks also occupy cold, deep habitats, move slowly, and remain metabolically economical, yet their lifespans are far shorter. The difference appears to reach into the architecture of the cells themselves. A chromosome scale genome, assembled by researchers led by the University of Tokyo between 2024 and 2025, contains about 5.9 gigabases of genetic material, placing the Greenland shark among the animals with exceptionally large genomes. Within that vast sequence, researchers identified multiple copies of 81 genes associated with repairing damage to deoxyribonucleic acid. That redundancy matters because every cell accumulates molecular injuries simply by existing. Chemical reactions can damage genetic material. Replication can introduce errors. Environmental stress can add further wear. A repair system with repeated copies of important genes provides more biological machinery for detecting and correcting those problems before they become permanent changes. The genome also contains an expanded nuclear factor kappa B signaling system, a network involved in immune responses and inflammation. In many animals, inflammation is useful in the short term, but damaging when it persists for years. The Greenland shark appears to have an unusual genetic architecture surrounding this process, potentially helping it manage the long biological timescale over which small injuries and immune reactions could otherwise accumulate. Another piece involves tumor protein 53, often described by scientists as a guardian of the genome. Alterations in this system appear to strengthen the shark's ability to suppress abnormal cell growth. Documented cancers in Greenland sharks are extraordinarily rare, although the scarcity of examined animals means scientists must remain cautious about interpreting that absence. Taken together, these features suggest that longevity here is not the result of one magical mechanism. It is a collection of small advantages operating together: an exceptionally slow metabolism, growth measured across generations, efficient cellular maintenance, unusual control of inflammation, and strong defenses against abnormal cells. The result is a body that does not rush through life. Damage arrives, as it does in every living organism, but the machinery responsible for dealing with it appears to work on a time scale suited to an animal that may remain alive for centuries. Aging a Greenland shark begins with accepting that the usual clock is missing. In many fish, scientists can examine structures that grow in repeating layers, much like rings in a tree. Each layer can preserve a record of another year. The Greenland shark offers no such convenient archive. Its cartilaginous skeleton does not produce annual growth rings that can be counted with confidence, leaving researchers without the ordinary calendar written into bone. The answer came from a structure that begins forming before the animal ever enters the ocean. Deep within the eye is a lens made partly from proteins called crystallins. These proteins are produced during embryonic development and then remain in place for the rest of the shark's life. They are not continually replaced in the way many other tissues are. Instead, the oldest material remains sealed within the center of the lens, preserving a chemical signature from the earliest stage of existence. That makes the lens something like a biological time capsule. As the embryo develops, the environment leaves an isotopic imprint in the material being formed. Once those proteins become enclosed inside the lens, the signal remains remarkably stable. The outside world can change for centuries, seasons can pass above the animal, and generations of humans can come and go, while that tiny region inside the eye retains evidence from the beginning. In 2016, marine biologist Julius Nielsen and his colleagues at the University of Copenhagen used this property to investigate the ages of 28 female Greenland sharks caught unintentionally in fisheries. The animals ranged from about zero point 0.8 meters, about 2.7 feet, to 5.0 meters, about 16.5 feet, in length. Instead of looking for rings, the researchers examined the radiocarbon contained within the nuclei of their eye lenses. Radiocarbon provided an unusual reference because its concentration in the ocean changed dramatically during the 20th century. Atmospheric nuclear weapons testing during the Cold War released large amounts of radiocarbon into the atmosphere, producing a recognizable increase that eventually entered marine food webs. This became known as the nuclear bomb pulse, a chemical marker that scientists could trace through organisms exposed to the changing environment. The youngest sharks in the study carried evidence of this modern signal, helping establish a relationship between their lens chemistry and known historical time. Larger animals showed something very different. Their eye lenses contained no indication that they had experienced the bomb pulse during their early lives. Their biological clocks reached back beyond the beginning of that unmistakable human signal. The largest shark, measuring 5.0 meters, about 16.5 feet, produced the most extraordinary estimate. Her age was calculated at 392 years, with an uncertainty range extending from 272 to 512 years. That means she may have entered the ocean sometime between 1504 and 1744. Even the lower estimate reaches farther into the past than the confirmed lifespans of other vertebrates traditionally regarded as exceptionally long-lived. A Greenland shark, born around the beginning of the 16th century, could have been swimming beneath northern waters while the Renaissance was still unfolding across Europe. The precision of the method is important because the researchers were not simply guessing from body size. The estimate came from chemistry locked inside a structure that had been forming since the animal's earliest development. The shark carried its own record of time within its eye, and that record reaches into an era when modern nations had not yet taken their familiar shapes, long before industrial society transformed the oceans. For a creature whose movements are measured in such small increments, its lifetime stretches across an almost unimaginable portion of human history. Imagine placing a single human lifetime beside the life of a Greenland shark, then extending the comparison until generations begin to overlap. A century for us can contain childhood, adulthood, old age, and the memory of an entire family. For this animal, a century can pass while its body is still approaching maturity. A shark, born in 1624, near the lower boundary of the oldest age estimate, entered the ocean during the reign of Charles I. 4 years later, the Mayflower crossed the Atlantic. The young shark would have been growing beneath northern waters. While European settlements were taking shape across a continent it could never have known. Isaac Newton was born in 1643. When he published his laws of motion in 1687, a Greenland shark born decades earlier could already have spent more than half a century alive. The animal's lifespan was unfolding alongside the emergence of ideas that would eventually reshape how humanity understood motion, gravity, and the physical world. By 1775, when the American Revolution began, a Greenland shark approaching 150 years of age could have been reaching the threshold of sexual maturity. Human political history moved through revolutions, wars, and new nations within a single lifetime. The shark's biology advanced at another scale entirely. The difference becomes especially tangible with the female eventually found off Cornwall, whose lifetime crossed an entire century of human transformation. She was approximately 100 years old when she died, having entered the ocean around 1922. That was the year the Soviet Union was formed and insulin was first used to treat diabetes. The British Empire stood near the greatest geographic extent it would ever reach. Commercial aviation was still young. Mount Everest had not yet been climbed, humans had not broken the sound barrier, and the moon remained untouched by any spacecraft or human foot. All of those milestones arrived during her lifetime, yet none represented a moment of biological adulthood for her. While cities expanded, borders changed, aircraft crossed oceans, and humans eventually walked on the lunar surface, her body continued through the same gradual process of growth that had begun before any of those events existed. The pathologists who eventually examined her were standing over an animal whose personal history reached back to the beginning of modern radio, early automobiles, and the first decades of powered flight. Still, by the standards written into her species, she had not reached reproductive maturity. She was ancient by our measure, yet biologically young. Female Greenland sharks generally become sexually mature at around 150 years of age. That places the Cornwall shark roughly 50 years short of the point at which she might have begun reproducing. Her century of existence had produced no offspring. The distinction matters because longevity and reproduction are linked in an unusual way here. A life can stretch across an enormous span of human history while remaining biologically unfinished. Her eye lens preserved another version of this history. The isotopic signature formed during her early development remained embedded within the lens for approximately a century, carrying chemical information from the ocean of 1922. The record was not written in diaries, monuments, photographs, or archives. It existed inside living tissue, protected by the structure of her own eye. That makes the passage of time feel unusually physical. Human history leaves traces in buildings and documents, while the Greenland shark carries its earliest history within itself. A century did not merely happen around her. It accumulated inside her, molecule by molecule, while the world above the water transformed into something almost unrecognizable. A seal can cross water at a speed the Greenland shark could never hope to match. Harp seals can reach nearly 20 kilometers per hour, about 12 miles per hour, while the shark generally cruises at less than 2 kilometers per hour, about 1 mile per hour. Yet, seal remains appear repeatedly in Greenland shark stomachs. The contradiction disappears only when speed stops being the measure of the hunt. The shark does not need to chase a seal across open water. Its advantage begins with where it approaches from. A Greenland shark can remain beneath its prey, moving upward through the water column at a pace that would seem almost incidental. Seals resting near the surface may be watching for faster predators around them, while the shark arrives from a direction where sight provides little warning. Electroreception becomes important here. Tiny sensory organs around the shark's head can detect electrical fields produced by living tissue, including the activity of muscles and the beating heart. The signal does not need to travel far to matter. At close range, a living seal creates a biological signature that exists whether the shark can see its prey or not. Combined with smell and the shark's ability to move with very little energy expenditure, this creates a hunting strategy based less on pursuit than on position and timing. Stomach contents provide the evidence that the strategy works. Greenland sharks have been found carrying fish, squid, skates, and other sharks, along with seals. Historical records describe even stranger meals, including polar bears, reindeer, moose, and horses that had entered the sea or drowned near ice edges. These accounts must be treated carefully because a stomach reveals what was consumed without always revealing how the animal obtained it. One 19th century record even described a human arm being recovered from a Greenland shark. The circumstances surrounding that report were never established with enough certainty to explain the event, and it remains part of the historical record rather than a clear observation of deliberate hunting. That distinction matters because this feeding behavior has almost never been witnessed directly. No person has watched a Greenland shark capture a seal in the wild. No camera has followed the complete encounter from approach to feeding. Much of what scientists understand comes from the combination of stomach contents, anatomy, sensory biology, and the conditions in which these sharks live. For years, another piece of evidence seemed to connect the species to seals. Carcasses on Sable Island, off Nova Scotia, were sometimes found with distinctive spiral wounds, and Greenland sharks were proposed as the cause. The idea was intriguing, but later work did not establish those wounds as a reliable signature of shark attacks. The origin remains debated, with boat propellers among the proposed explanations. The more consistent picture is much less theatrical. A Greenland shark does not need to overpower a healthy seal in a prolonged chase. Its metabolism allows it to wait, and its sensory systems allow it to locate living prey without relying entirely on vision. If an opportunity appears at close range, the difference between 1 mile per hour and 12 miles per hour becomes almost irrelevant. That patience also changes the meaning of feeding frequency. An animal that spends so little energy at rest does not need to hunt continuously. Meals may be separated by days or weeks, depending on prey availability, body condition, and temperature. The shark can exist between opportunities without constantly searching. The female found near Cornwall appears to have reached the end of that long economy in poor condition. Her body was underweight and nutritionally depleted, suggesting that feeding had already been disrupted before she entered the shallower water. Whatever opportunities had once sustained her were becoming harder to reach. Her stomach would eventually preserve a small part of that story, showing that the decline had begun before her final journey toward the surface. There is a strange boundary between the Greenland shark and the animals around it, and it is written into the chemistry of its flesh. Tissue that allows this shark to function under immense pressure becomes dangerous when that same body is brought into the world above. One of the most important compounds is trimethylamine N oxide, a small molecule distributed throughout the shark's tissues, including its blood and muscles. At great depth, pressure presses against every cell. Proteins can lose their proper shape, enzymes can become unstable, and the delicate molecular structures required for life can begin to fail. Trimethylamine N oxide helps prevent that collapse by stabilizing proteins, allowing the shark's chemistry to continue working where pressure would otherwise interfere with the machinery of life. The protection comes with a cost once the shark's tissues enter a mammalian body. When untreated Greenland shark tissue is eaten by mammals, compounds associated with that chemistry can interfere with the nervous system. Historical accounts from Greenland describe working sled dogs becoming unsteady after consuming raw shark meat. Animals could stagger, lose coordination, circle without control, and remain incapacitated for hours. Large quantities could be fatal. Humans are vulnerable as well. Untreated flesh can cause vomiting, disorientation, and severe loss of motor control, with poisoning becoming more serious as exposure increases. The shark also carries unusually high concentrations of urea throughout its tissues. Urea is a normal waste compound in many animals, but in this case it exists alongside trimethylamine N oxide as part of the biochemical adaptation that allows the shark to inhabit deep water. The result is a body that has solved one environmental problem by creating another. Chemistry that protects delicate proteins from pressure does not become harmless simply because the animal has been brought into shallower water. For people who encountered these sharks as food, another solution had to be found. Icelandic communities developed that solution through fermentation. The traditional preparation of hakarl begins by removing the shark's head and internal organs, then placing the body beneath stones in coarse gravel. The pressure allows fluids to drain while fermentation changes the chemistry of the flesh. After roughly 6 to 8 weeks, the meat is removed and hung to dry for several additional months. During this long transformation, compounds responsible for the raw shark's toxicity are broken down into forms that can be consumed more safely. The finished food is famous for its powerful ammonia-like odor produced by the chemical changes occurring during fermentation and drying. Hakarl is traditionally served with brennivine, a strong Icelandic spirit whose name is often translated as Black Death. The pairing belongs to a culinary tradition shaped by necessity rather than novelty. Before modern preservation methods, an animal that could not safely be eaten fresh could still become food if its chemistry was patiently altered. The practice reaches back through Icelandic history for centuries, connecting modern meals with generations of people who learned through experience that this particular shark required time before it could enter the human diet. Somewhere in that history, careful observation became tradition, and tradition eventually preserved the knowledge long enough for modern chemistry to explain it. There is something fitting in the fact that the Greenland shark carries its own environmental solution within every cell. Its body survives pressure by changing the balance of molecules inside itself, while the people who harvest it must change that chemistry again before eating it. Deep below the surface, the same compounds that preserve the shark's life are part of what makes its flesh unsuitable for ours. Above the water, patience becomes the final step in making that ancient chemistry edible. For much of its life, the Greenland shark carries another animal directly against its vision. Omatocoida elongata is a parasitic copepod, a small crustacean that attaches to the surface of the eye. Its body can extend outward from the cornea like a pale thread, remaining anchored while the shark continues moving through the ocean. The attachment is remarkably intimate. The parasite grips the corneal surface and feeds on tissue there, remaining with its host for long periods. Among adult Greenland sharks, infection is extremely common, with many individuals carrying the copepod on one or both eyes. Over decades, repeated irritation and scarring can make the normally transparent cornea increasingly opaque. For years, that appearance shaped the scientific picture of the animal's vision. A cloudy eye seemed to suggest a damaged sensory system, especially in a species already living where very little light exists. The parasite became associated with the idea that Greenland sharks were largely, perhaps almost completely, blind. Then the eye itself was examined more closely. In January 2026, researchers from the University of Basel and the University of California, Irvine published a study in Nature Communications that examined the Greenland shark visual system using anatomical, genetic, biochemical, and functional evidence. The animals examined included adults estimated to be more than a century old. Their retinas showed no obvious signs of the progressive degeneration that might be expected after such an extraordinary lifespan. Inside the retina, the researchers found densely packed, elongated rod cells specialized for detecting very small amounts of light. Other essential retinal cell types were also present, preserving the circuitry needed to process visual information. The evidence suggested that the shark had not abandoned vision. Instead, its eyes had become highly specialized for the faint blue wavelengths that remain available in deep water. The distinction between the cornea and the retina is important. The cornea is the transparent outer surface through which light enters the eye. The retina lies farther inside, where specialized cells convert photons into electrical signals that can be processed by the nervous system. A cloudy surface does not automatically mean that the sensory machinery behind it has failed. Researchers also tested whether light could still pass through parasitized corneas. The shark corneas examined transmitted substantial amounts of light, including within the blue portion of the spectrum that is especially important in deep water. The parasite may obstruct part of the visual field, but it does not appear to make the entire visual system useless. There was an even more direct indication. Video footage showed Greenland sharks moving their eyes toward sources of illumination, a behavior consistent with active visual tracking. The movement is subtle, but it provides a living response rather than a conclusion drawn only from preserved tissue. Their vision also appears to be maintained at the molecular level. Genes involved in detecting dim light remain functional and active, while many genes associated with bright light vision have been reduced or lost. The retina also shows strong expression of genes involved in repairing cellular damage, suggesting that the same biological maintenance systems supporting extreme longevity may help preserve the eye itself. The Cornwall female had cloudy corneas in both eyes, consistent with long-term parasitic damage, yet that finding cannot be treated as evidence that her retinas had failed. The parasite had altered the window through which she saw the world, but the deeper machinery of vision appears to have remained intact. That makes the eye of the Greenland shark an unusual record of endurance, carrying an organism that damages its surface while preserving a sensory system capable of remaining functional across more than a century of life. For all the precision that science had gained about the Greenland shark, one part of its existence remained almost entirely beyond observation: the end. Researchers could estimate its age from the chemistry of its eyes, measure its metabolism, study its genome, and reconstruct much of its feeding ecology. Death was different. There was almost no body left behind to study. A Greenland shark that dies in its usual habitat does so far below the surface, in water where human observers cannot simply descend and watch the process. At depths approaching 2,400 meters, about 8,000 feet, darkness surrounds the animal completely, while extreme cold slows decomposition. Scavengers begin their work, tissues disperse, and currents separate the remains. By the time anything could reach the site, much of the evidence that might explain the death would already be gone. Even when these sharks leave the deep, they rarely arrive where scientists can examine them. Strandings are extraordinarily uncommon, particularly in places with established marine pathology programs. Before 2022, only 3 Greenland shark strandings had been recorded in waters around the United Kingdom. Each represented a rare encounter with an animal normally separated from human observation by an enormous vertical distance. One of those events occurred in Northumberland in 2013. A Greenland shark reached the British coast, creating an opportunity to examine a species that had almost never been available to marine pathologists. Yet, no detailed post-mortem examination was conducted. The body disappeared without producing the kind of forensic record that could reveal what had happened inside it. The absence was striking because the United Kingdom already possessed an extensive system for investigating marine deaths. The Zoological Society of London's Cetacean Strandings Investigation Program had examined thousands of stranded marine animals since 1990, including whales, dolphins, porpoises, turtles, and basking sharks. Its pathologists had developed years of experience reading disease, injury, starvation, infection, and human impact from the physical remains of animals found along British shores. The Greenland sharks simply had not appeared on their tables. Meanwhile, knowledge of the living animal was becoming increasingly detailed. Radiocarbon analysis had transformed estimates of age. Metabolic studies had revealed how little energy the shark uses at rest. Genetic research was beginning to expose the cellular systems associated with its extraordinary longevity. Observations of stomach contents and sensory anatomy offered increasingly sophisticated ideas about how it feeds. Yet all of that knowledge described an animal while it was functioning. There was almost no comparable evidence for the moment when those systems began to fail. Scientists could infer that age, disease, injury, starvation, parasites, environmental conditions, or some combination of factors might eventually end a Greenland shark's life. But inference is different from observation. Without a body examined soon after death, there was no way to distinguish a plausible explanation from a demonstrated cause. That created an unusual imbalance in the scientific record. The longest-lived vertebrate known to science had been studied across centuries of biological time, yet the physical process of its death remained largely invisible. There were no established patterns of organ failure, no reliable catalog of diseases, and almost no pathological baseline against which a sick individual could be compared. The missing evidence was not hidden in some inaccessible archive. It was disappearing naturally beneath layers of cold seawater every time another ancient shark died beyond human reach. Each death became part of the deep ocean before science could arrive, leaving only fragments of a story that no observer had witnessed from beginning to end. Then, in March 2022, one of them reached the shore. On the morning of March 13th, 2022, the edge of Newlyn Harbor held something that did not belong there. A dog walker noticed a large shark lying in the surf near Penzance, on the southwestern coast of England. It was a Greenland shark, a species normally separated from this coastline by immense distances and deeper water. She measured 3.96 meters, about 13 feet, from nose to tail, and weighed 285 kilograms, about 628 pounds. She was female, and despite her considerable size, she had not yet reached sexual maturity. Her body carried signs that something had already gone wrong. The fins were damaged, and the tissue around them showed evidence of physical trauma. There was another detail that seemed almost out of place on a beach. Inside her stomach was silt, fine sediment from the seafloor. It was not simply material that had entered her body after death. She had swallowed it while still alive, taking water and sediment into herself during the struggle that brought her toward shallower ground. The detail would later become one of the most important pieces of evidence in reconstructing her final movements. For the moment, though, there was no laboratory, no examination table, and no time to begin the investigation properly. The returning tide lifted the shark and carried her away from the shore before researchers could secure the body. She disappeared back into the same sea that had brought her there, leaving only photographs, observations, and the possibility that the opportunity might be lost. A search followed along the coast. Recreational boats moved through the waters around Newlyn Harbor, while coastal researchers and personnel from the Zoological Society of London looked for signs of the missing animal. The search was unusually difficult because a carcass that large could drift, sink, or be carried beyond the harbor by currents and changing tides. 2 days later, on March 15th, a tourist boat located her floating in the water off Newlyn Harbor. By then, she was dead. Her body was recovered, preserving an opportunity that had almost disappeared with the tide. The recovery brought her into the care of the Cornwall Marine Pathology Team, where veterinary pathologist James Barnett and his colleagues prepared to examine an animal unlike any they had previously encountered. The examination took place on March 16, 2022, as part of the Zoological Society of London's Cetacean Strandings investigation program. What lay before them was not simply a large shark, it was a specimen from a species whose deaths had almost never been available for detailed forensic study. The body could be measured, opened, sampled, and examined at a level that had never before been possible in the United Kingdom. The physical scale of the animal also carried a strange historical weight. She had lived for approximately a century before arriving on this Cornish shore, yet everyone in the room was seeing her body only after her life had ended. The pathologists could not observe the years she had spent growing or the precise moment when her health began to decline. They could only read the evidence that remained in her tissues. Rob Devil, the project lead for the investigation program, later described the animal as almost certainly the oldest specimen he had ever personally examined. Around her, instruments and sampling equipment turned an unexpected beach discovery into a scientific record. For the first time in the United Kingdom, a Greenland shark had been brought within reach of marine pathology, and her body was ready to reveal what the ocean had hidden. The first clues were not hidden deep inside her body. They were written across the skin and fins, in places where pressure, abrasion, and impact had left their marks. Around the head and pectoral fins, pathologists found hemorrhages within the soft tissues. The fins were damaged in a pattern consistent with repeated contact against an abrasive surface. Inside the stomach, the silt remained, a trace of the seafloor carried into an organ that should never have contained it. Those hemorrhages were especially important because bleeding into living tissue requires circulation. The damage had occurred while blood was still moving through her body. She had not simply drifted onto the shore after death. Her tissues recorded a period of physical struggle, consistent with an animal still moving as it encountered the seabed and later the beach. The evidence suggested that her final journey involved active contact with the bottom rather than a body passively carried there. Then the examination reached the skull. When the pathologists opened it, the brain did not have the appearance expected from healthy tissue. It was visibly discolored and congested, with changes apparent before any microscopic preparation or laboratory testing could begin. Surrounding it was cerebrospinal fluid, the clear liquid that normally cushions the brain and spinal cord. In this case, the fluid was cloudy. Cloudiness in cerebrospinal fluid is an important warning sign because it can indicate an accumulation of inflammatory cells, proteins, and other material associated with infection. The observation shifted the investigation from physical trauma toward disease. Something had been happening inside the skull before the shark reached the coast. Microscopic examination provided the next layer of evidence. Thin sections of brain tissue were examined under magnification, allowing the pathologists to look beyond what could be seen with the naked eye. The tissue showed inflammation consistent with bacterial meningitis, an infection involving the membranes surrounding the brain and spinal cord. The laboratory work then identified the organism involved. Bacteria belonging to the genus Pastorella were isolated directly from the cranial fluid. The finding connected the visible inflammation with a specific infectious process. Together, the gross examination, microscopic tissue changes, Abnormal cerebrospinal fluid and bacterial isolation produced a diagnosis that had never before been confirmed in this species. The Greenland shark had bacterial meningitis caused by Pasteurella infection. That diagnosis gave new meaning to the circumstances of her stranding. The membranes around the brain are not passive coverings. They are part of a system that protects an organ responsible for coordinating movement, orientation, and responses to the surrounding environment. When those tissues become inflamed, neurological function can deteriorate. In vertebrates, meningitis can produce disorientation, impaired spatial awareness, and difficulty maintaining normal directional movement. For an animal that normally lives in a three-dimensional world of changing currents and depth, those impairments could have profound consequences. A healthy Greenland shark does not need to move rapidly to remain within its environment. Its body is adapted to conserving energy while maintaining position and navigating through immense stretches of water. But navigation depends upon a functioning nervous system. The evidence, therefore, suggested something more subtle than a simple collision with the coast. Her damaged fins and hemorrhaged tissues showed what happened when her body encountered the seabed and shore. The infected brain offered a possible explanation for why she had reached that place at all. James Barnett, the veterinary pathologist who examined her, concluded that the poor condition of the shark, together with the hemorrhaging around the pectoral fins and the silt inside her stomach, indicated that she may have stranded while still alive. Her body had preserved the sequence in fragments. Infection had altered the brain. Disorientation may have followed. Then the animal that had spent a century navigating the deep ocean lost the ability to remain within the world that had sustained her. A body adapted to the deep ocean does not simply cross the boundary into shallow water without paying a physiological price. For the Greenland shark, the ascent from its usual depths would have meant passing through a pressure gradient that changed with every part of the journey. Her normal habitat lies roughly 1,800 to 2,400 meters, about 6,000 to 8,000 feet, beneath the surface, where the surrounding water presses against her with extraordinary force. At 2,400 meters, about 8,000 feet, the pressure approaches 240 atmospheres, roughly 240 times the pressure at sea level. That pressure is not concentrated at one point. It surrounds the animal continuously, shaping the physical conditions in which its cells, membranes, and proteins have evolved to function. Her tissues were built within that environment over an immense span of evolutionary time. Then the pressure began to fall. Every rise through another 30 meters, about 100 feet, removed another layer of water from above her. The change would have been gradual, but relentless. The deeper architecture of her body was being carried into conditions for which it had never been designed. Nothing about the ascent needed to be deliberate. A damaged nervous system could turn an ordinary movement into a path with no reliable correction. Temperature added another strain. Coastal waters around Cornwall in March are commonly around 9 to 10 degrees Celsius, about 48 to 50 degrees Fahrenheit. The Greenland shark is adapted to water near freezing, with its preferred range roughly -2 to 1 degree Celsius, about 28 to 34 degrees Fahrenheit. Moving into warmer water increases metabolic demand in a body whose entire strategy depends upon conserving energy. Reactions accelerate. Tissues require more resources, and the thermal conditions become progressively less familiar to an animal shaped by cold. Depth also brought a change that was not physical alone. For a shark accustomed to profound depths, illumination would normally be almost absent. The photic zone begins hundreds of meters below the surface, around 200 meters, about 650 feet, where the last traces of filtered daylight can persist. Beyond that boundary, darkness dominates. A century spent in the deep ocean would have made shallow coastal light an entirely different sensory world. Yet vision was only one part of the problem. Greenland sharks possess electroreceptive organs capable of detecting the faint electrical fields produced by living animals. Their sense of smell is also important for locating biological signals carried through seawater. Both systems depend upon a functioning nervous system capable of interpreting information and coordinating movement. Meningitis affecting the brain could interfere with that coordination, turning useful sensory information into signals that were difficult to organize into a stable sense of direction. As she rose, the ocean itself changed around her. Pressure diminished, temperature climbed, light appeared. The chemistry of the surrounding water remained seawater, but its physical character was becoming increasingly foreign. Depth cues that had shaped her movements for a lifetime were disappearing, while the sensory landscape grew more complicated. Near the surface, the water was not simply shallower, it belonged to another part of the planet. The shark was entering a zone where waves moved the body differently. Currents interacted with the coastline, and sunlight could reach the water from above. Her damaged brain could no longer reliably interpret the environment or guide her back toward deeper conditions. The ascent, therefore, did not resemble a purposeful journey toward land. It was the physical consequence of losing the neurological ability to remain where her body had always belonged. She was not choosing the surface. Her failing brain had removed the information that once kept the surface away. The sand tells part of the story, but the stomach tells it more precisely. Inside her was sediment from the seafloor, material that could only have entered her digestive tract while she was still underwater. She had swallowed it before reaching the beach, drawing seawater through her gills as she moved along the bottom. That single trace places her alive on the seabed, close to the beginning of the final sequence. A shark does not possess a swim bladder to support its body against gravity. Instead, Greenland sharks rely heavily on a large, oil-rich liver that contributes substantially to buoyancy. In the water, the surrounding pressure and density distribute the animal's enormous mass. On land, that support disappears. Her body would have rested directly against the sand, concentrating pressure through the organs and tissues beneath her. Breathing would have become another problem. Her gills were adapted to cold seawater, where dissolved oxygen could support the small metabolic demand of her deep ocean life. Coastal water around Cornwall in March was considerably warmer than the conditions she normally inhabited. Warmer water holds less dissolved oxygen, while the effort required to move and exchange water across the gills becomes increasingly important when an animal is already compromised. On the beach, there was no longer a stable column of water surrounding the gill structures. Every breath became more difficult as the conditions deteriorated. The pectoral fins carried their own record of those moments. They were damaged in ways consistent with contact against an abrasive surface, with torn tissue suggesting that movement continued while her body was being dragged or pressed against the bottom. This was not the mark of an animal that had simply arrived after death. Her muscles were still working. Her body was still responding to its surroundings. Around those damaged fins, the pathologists found hemorrhaging within the soft tissues. Blood entering injured tissue requires circulation, and circulation requires a functioning heart. The injuries therefore belonged to the period when she was still alive. Her final movements were being recorded within her own body as physical changes that remained after movement had ceased. Yet, she had not reached the coast in good condition. The shark was underweight and nutritionally depleted, and the infection found within her brain had already been established before the stranding. Meningitis can disrupt normal neurological function, and in her case, the disease likely reduced her ability to feed and navigate effectively. The decline may have unfolded over days or weeks, gradually removing the reserves that had sustained an animal capable of surviving for a century. Then came the strange interruption in her final hours. She was discovered near Newlyn Harbor, examined briefly, and then taken away again by the returning tide before a full investigation could begin. For 2 days, her body remained somewhere between the harbor and open water. The exact path she followed during that interval cannot be reconstructed, but the physical evidence shows that the transition from deep ocean to shore was not instantaneous. An animal that had spent a lifetime moving through immense depths ended its life against a Cornish beach, where gravity replaced buoyancy, warmer water replaced the cold, and the seabed gave way to sand. Her body carried the marks of each stage, preserving a physical chronology that could still be read after the living animal was gone. The sediment recorded where she had been, the damaged fins recorded movement, the hemorrhages recorded life within injured tissue, and the infected brain explained how the animal that once navigated the deep ocean could have lost its way toward the shore. There is something almost ordinary about the organism found inside her. Pastorella is not a creature of abyssal darkness, nor a specialized pathogen shaped exclusively by Arctic waters. It is a widespread group of bacteria encountered across marine and terrestrial environments, living among animals that share the same planet as the Greenland shark, but occupy radically different worlds. Among its familiar hosts are rabbits, cattle, cats, and dogs, where particular Pastorella species can cause respiratory disease or infections associated with wounds. Humans can encounter it as well, especially after bites from domestic animals. In many ordinary circumstances, these infections are treatable. The bacterium belongs to a microbial world that is surprisingly familiar, even though the animal it infected was anything but. Pastorella has appeared in other sharks. Researchers have documented infections in captive lemon sharks, while related findings have occurred in beached basking sharks. Those cases matter because they establish that the genus can interact with shark tissues. Yet the Cornwall discovery was different. Before this examination, a confirmed Pastorella infection had not been documented as the cause of meningitis in a Greenland shark, a species adapted to the cold northern ocean and a life spent far below the surface. How the bacterium entered her body cannot be reconstructed from the post-mortem evidence. It may have crossed damaged tissue, entered through the gills, arrived through contaminated prey, or reached the shark by another route that left no recognizable trace. The examination could identify the infection and its consequences, but not the precise moment when the bacterium gained access to her body. That uncertainty becomes more striking when placed beside the biological defenses already known in this species. The Greenland shark possesses an unusually large collection of genes associated with repairing damaged deoxyribonucleic acid. Researchers have also identified an expanded nuclear factor kappa B signaling system involved in immune regulation and inflammation, along with changes involving the tumor protein 53 pathway, a major regulator of cellular damage and tumor suppression. An animal with such elaborate mechanisms for maintaining its cells had still succumbed to a bacterium familiar from much more ordinary infections. That does not mean the Greenland shark was invulnerable until Pastorella arrived. Long life is not the same as perfect immunity, and cellular repair systems do not guarantee protection against every infectious disease. The infection may have overwhelmed defenses that were already compromised, or it may have exploited a weakness that cannot be identified from a single carcass. The available evidence does not establish that pollution, warming waters, fishing pressure, or another human influence caused her infection or weakened her immune system, nor does it reveal when her defenses first began to fail. This distinction matters because the temptation to connect an unusual death with a changing ocean is strong. Yet science cannot replace missing evidence with a compelling story. In this case, the responsible conclusion is narrower and more interesting. A common bacterium caused a disease that had never before been confirmed in this species, and the circumstances that allowed that infection to become fatal remain unknown. For a creature capable of surviving across centuries, the final cause of death was not some mysterious force unique to the deep ocean. It was a microscopic organism found in familiar animals, acting within a body whose defenses had evolved over immense stretches of time, over ages. And because only one Greenland shark had been examined this way, there was no second case waiting nearby to tell scientists whether her death was an anomaly, a vulnerability shared by the species, or the first visible trace of something larger. For more than a century, the Greenland shark had been one of the great absences in marine natural history. Scientists could estimate its age, examine its genetics, study its metabolism, and document the strange chemistry that allowed it to inhabit deep northern waters. Yet, when one finally died in a place where people could reach the body, something unprecedented became possible. Its death could be examined rather than imagined. The Cornwall shark, therefore, became more than a stranded animal. Her post-mortem examination produced the first detailed forensic record of how a Greenland shark died, creating a reference that had never existed for this species. Before this case, there was no published necropsy report for a Greenland shark in the United Kingdom, and detailed documentation of natural death in wild Greenland sharks elsewhere was extraordinarily scarce. The gap was not caused by a lack of interest. The species simply lived and died beyond the reach of ordinary investigation. The scale of that absence becomes clearer through the work of the Zoological Society of London's Cetacean Strandings Investigation Program. Since 1990, its specialists have examined thousands of marine animals, including whales, dolphins, porpoises, turtles, and basking sharks. Nearly 4,500 necropsies had been conducted, yet the Greenland shark had never been placed on their examination table. A species that had existed around northern oceans for millions of years had somehow remained almost completely absent from the forensic record. That changed with the examination of the Cornwall female. The findings were documented, analyzed, and submitted for peer review before being formally published in the scientific literature. Once published, her case ceased to belong only to the people who stood around her body in Cornwall. It became a permanent scientific reference, available to researchers who may someday encounter another Greenland shark in circumstances that can be compared with hers. Rob Devil, who leads the Strandings Investigation Program, described the event as an extraordinary opportunity to gain insight into a species whose life and death remain poorly understood. The word extraordinary carries unusual weight here because it came from someone accustomed to examining marine animals after death. For a scientist who has spent years studying thousands of stranded bodies, this was not simply another unusual carcass. The body itself became the evidence. The inflamed tissues recorded an infection that had reached the membranes surrounding the brain. Pasturella was recovered from the cranial fluid. Hemorrhaging preserved the signs of physical struggle. Silt inside the stomach showed that she had been alive near the seabed before reaching the shore. Each finding occupied a small place within the body, yet together they created something much larger, a record of events that science had never before been able to observe directly in this species. That distinction matters because a scientific record does not require a complete explanation. It begins with an observation that can be preserved, described, challenged, and used by others. Her examination established a baseline for pathology in a species whose natural history has been reconstructed largely from living animals, tissue samples, models, and rare encounters. Future cases may confirm parts of her story, contradict others, or reveal diseases that were invisible in Cornwall. But without a first case, there would be nothing against which those discoveries could be measured. There is something profound in that first record. The Greenland shark had survived an environment almost entirely inaccessible to human observation, carrying a history measured in generations rather than ordinary lifetimes. Then one individual reached the surface world and left behind the first detailed account of her species' death. Her body became a document written in tissue, fluid, damaged fins, and sediment, and the scientific literature now has its first page. For a century, the world available to her eyes had been almost entirely without light. That does not mean those eyes had stopped working. New evidence published in January 2026 has changed that part of her story, revealing that the Greenland shark carried a functioning visual system through an extraordinary span of time. Researchers examined the eyes of 10 Greenland sharks, ranging from 100 to 134 years old, including animals within the same age range as the Cornwall female. Their retinas showed no detectable degeneration in the photoreceptor cells responsible for receiving light. The rods, specialized for detecting faint illumination, remained structurally intact despite decades spent in deep water. Age had not simply erased the machinery of vision. Video footage provided another piece of the evidence. Living Greenland sharks were observed responding to light sources, directing their eyes toward illumination. Their movements showed that the visual organs were not merely preserved anatomical relics. They remained connected to a functioning sensory system capable of registering changes in the world around the animal. That finding changes the meaning of the Cornwall shark's ascent. Earlier assumptions had treated the parasite-damaged surface of her eyes as evidence that she was probably blind. The newer evidence separates two very different things. A cloudy cornea can scatter and reduce incoming light, while an intact retina can continue converting whatever light reaches it into signals for the nervous system. Her eyes may have been impaired, but there is no reason to assume they were useless. As her body rose through the water column, the darkness would have begun to thin gradually. Around 198 meters, about 650 feet, faint filtered sunlight can begin reaching the ocean below the brighter surface layers. At roughly 61 meters, about 200 feet, daylight becomes much more apparent. Near 9 meters, about 30 feet, the water around Cornwall in March would have carried a level of illumination utterly different from the environment in which her visual system had spent a century operating. For an animal adapted to extremely low light, the transition would have been unlike an ordinary change from night to day. Her retinal cells were specialized for collecting scarce photons, making the most of tiny quantities of available illumination. At the surface, far more light would enter the eye from every direction. Exactly how a Greenland shark experiences such a transition remains unknown, and the surviving evidence cannot reconstruct her final visual impressions. The biology can only establish that her eyes possessed the machinery to receive them, even as the rest of her nervous system was failing. Meanwhile, the infection affecting her brain would have been altering the systems that normally organize information from the senses. Light could reach her eyes while the neural pathways responsible for interpreting the world were becoming increasingly unreliable. The contrast is difficult to escape. Her body carried sensory equipment, refined for an environment almost untouched by sunlight. Yet her final movement brought that equipment into a place filled with it. She may have seen the pale surface approaching long before any human saw her. She may have registered brightness spreading through the water, shapes breaking across the illuminated upper layers, and the unfamiliar geometry of the coast. None of those details can be recovered from tissue or sediment. What the evidence preserves is something more fundamental. The old shark was not necessarily entering the light as a blind animal. Her eyes had survived the century. They were still capable of receiving the world, even when that world was no longer the one her body had evolved to inhabit. A Greenland shark does not simply live through time. It gathers pieces of the ocean into itself, carrying traces of the places it has traveled and the animals it has consumed. Every meal is also an encounter with chemistry. Pollutants absorbed by smaller organisms can pass into fish, squid, and other predators, becoming increasingly concentrated as they move upward through the food web. This process, known as bioaccumulation and biomagnification, gives a long-lived predator an unusual relationship with its surroundings. A contaminant released into seawater may remain present long after the original source has disappeared. When microscopic organisms encounter it, they can absorb it into their tissues. Small animals are eaten by larger ones, and those animals become food for larger predators. With each transfer, persistent compounds can become more concentrated. At the upper levels of the food web, the accumulated chemical burden can be far greater than the concentration that first entered the ecosystem. Greenland shark tissues have revealed several groups of industrial chemicals, including polybrominated diphenyl ethers, used as flame retardants, short-chain chlorinated paraffins, and polychlorinated biphenyls. The names sound distant from the cold waters where these sharks live, yet their persistence allows them to travel far beyond the places where they were originally manufactured or released. Polychlorinated biphenyls are especially revealing because their production was banned or heavily restricted in many countries decades ago, yet the compounds remain in sediments and marine ecosystems. They do not simply vanish when industry stops producing them. Some persist in the environment, circulate through food webs, and continue appearing in animals long after their original use has ended. For a shark born around 1922, that history would have unfolded across an extraordinary span. Industrial chemicals released during the 20th century could enter the ocean, settle into sediments, become part of marine food webs, and eventually reach a predator that had been alive for generations of human technology. Her body could therefore contain traces of an industrial world that existed before modern environmental regulation, preserved alongside the biological chemistry of an animal whose lineage belongs to far older oceans. The same principle applies to sharks living today. Their tissues are exposed to whatever persistent substances move through the ecosystems they inhabit, including newer industrial compounds, plastic-associated chemicals, pharmaceutical residues, and heavy metals. Some contaminants can remain in marine environments for years or decades. Others can be transformed into different compounds that may themselves persist or move through food webs. The ocean does not maintain a clean boundary between the past and the present. Chemistry released long ago can remain part of the biological world long after the people responsible for it are gone. A Greenland shark that survives for 2 or 3 centuries could therefore become something more than an old animal. Its tissues may preserve a layered chemical history of the ocean, with substances entering its body at different points across a lifetime that outlasts most human institutions. A shark alive near the end of the 23rd century could carry compounds introduced into northern marine ecosystems centuries earlier, mixed with whatever pollutants enter those waters during its own lifetime. For the Cornwall female, scientists cannot say whether accumulated contaminants contributed to the infection that ultimately killed her. There is no evidence strong enough to make that connection. Her toxic burden remains one variable among many, waiting for additional cases and better comparisons. Yet that uncertainty gives her body another dimension. The tissues examined after death were not only biological remains, they were also a record of the ocean she had inhabited, preserving traces of human activity inside an animal that had been alive before many of those chemicals were ever released. For generations beyond written memory, the relationship between Icelanders and the Greenland shark was shaped by necessity. The animal belonged to waters that could be dangerous to enter, yet its flesh could become food if handled with patience. Hackerel emerged from that practical relationship, and its history reaches back more than 1,000 years into the Viking Age and the early centuries of settlement in Iceland. Fresh Greenland shark meat cannot simply be prepared like ordinary fish. Its tissues contain unusually high concentrations of urea and trimethylamine oxide, compounds that protect the animal's cells in the deep ocean but make untreated flesh poisonous to people. Long before anyone understood proteins, pressure, or molecular chemistry, generations of Icelanders had discovered the solution through experience. The shark was gutted and beheaded, then placed into coarse gravel and covered with stones. There it remained for roughly 6 to 8 weeks, about 42 to 56 days, while its own weight and the pressure of the stones encouraged fluids to drain from the flesh. During that time, chemical changes reduced some of the compounds responsible for its toxicity. The meat was then removed and suspended in a drying shed, where it remained for several more months. What emerged was no longer raw shark, but a transformed food produced through a process refined across generations. The finished haakarl is famous for its powerful ammonia aroma. For someone encountering it for the first time, the smell can be overwhelming, arising from the same chemistry that makes untreated Greenland shark flesh unsafe. Traditionally, small pieces are accompanied by brennivin, an Icelandic aquavit whose name is often translated as Black Death. The strong spirit does more than provide contrast. Its intensity becomes part of the ritual, helping balance the remarkable sensory character of the fermented meat. There is another layer to this tradition that belongs to the shark itself. Some Greenland sharks captured for food during the past several centuries may have been extraordinarily old, potentially living for more than 300 years. An individual brought to shore could have entered the ocean before the lives of several human generations had unfolded, carrying within its body a history that reached far beyond the fishermen who eventually encountered it. That does not make haakarl a relic of some untouched past. The tradition survives today on a small scale, produced by specialized Icelandic makers rather than through a vast commercial harvest. Greenland shark fishing is regulated, reflecting the vulnerability of an animal whose biology makes it exceptionally slow to replace individuals removed from the population. The cultural practice, therefore, exists alongside modern knowledge that its raw material comes from a species unlike almost any other fish in the North Atlantic. What makes the tradition remarkable is not that people somehow conquered the shark's strange biology. They did the opposite. The preparation method acknowledges that biology, at every stage, the flesh must be drained, transformed, and aged before it can become food. The chemistry of the animal sets the terms, and human tradition learned to work within them. Across more than 1,000 years, that understanding became embedded not in laboratory measurements, but in repeated practice. People learned how long the meat needed to remain under stones, how long it needed to dry, and what the finished product should smell like. The result is a rare cultural record of coexistence with an animal whose biology demands accommodation rather than control, a relationship sustained because the shark itself established the rules. Its biology was never an inconvenience to be ignored. It was the foundation of the tradition itself, passed from one generation to another. For centuries, the Greenland shark was not simply encountered by people along the North Atlantic, it was pursued as a resource. Its extraordinary longevity, invisible behind the immediate value of its liver. Commercial hunting began as early as the 13th century and continued into the middle of the 20th century, creating a long period in which an animal capable of living for centuries was treated as a renewable catch. The reason was concentrated inside the shark itself. Its liver is enormous, sometimes representing up to 20% of total body weight, and packed with oil that could be rendered for practical uses. Before modern synthetic products became widely available, Greenland shark liver oil provided fuel for lamps, lubrication for machinery, and vitamin A. A single animal, therefore, carried several commodities within its body, making the slowest growing predator in northern waters valuable, precisely because of the chemistry that allowed it to survive its own extreme environment. During the 19th and early 20th centuries, the scale became substantial. Norwegian, Icelandic, Greenlandic, and Canadian fisheries sometimes removed more than 30,000 Greenland sharks in a single year. Each figure represented another subtraction from a population whose members could require many decades merely to reach maturity. The mathematics of such a fishery was profoundly mismatched with the biology of the animal. Long-lived species can withstand some losses when reproduction replaces individuals within a reasonable span. The Greenland shark has no such margin. Fishing pressure could preferentially remove the largest animals, including older females with the greatest reproductive value. A female that had survived for 150 years before contributing substantially to the next generation represented an accumulation of time that could not be restored by simply leaving younger sharks behind. Removing a 300-year-old female erased not only one animal, but centuries of survival that had already carried her through predators, scarcity, disease, and changing seas. The directed fishery eventually faded, but the threat did not disappear with the last major liver oil operations. Greenland sharks are still caught accidentally in fisheries targeting other species. In the Northwest Atlantic, the Arctic Ocean, and the Barents Sea, an estimated 3,500 individuals are taken as bycatch each year, entangled in nets or hooked on long lines, intended for fish such as halibut and cod. For a shark adapted to great depth, being hauled toward the surface introduces another physical problem. Pressure falls rapidly during ascent, changing conditions throughout the body in ways its physiology was never designed to experience. Even when a captured shark is returned to the sea, its chances of surviving the injury and physiological stress of capture and release remain poorly understood. The absence of precise survival data leaves another part of the mortality hidden beneath the surface. Historical catch records offer a broader measure of what these losses may have meant. Population reconstructions suggest that Greenland sharks declined by roughly 57 to 60% since the early 17th century, spanning more than 4 centuries of human contact. The species endured that history without disappearing, but endurance is not the same as recovery. In 2020, the International Union for Conservation of Nature changed the Greenland shark's status from near threatened to vulnerable. The designation placed a formal scientific weight on a pattern that had been accumulating for generations across northern seas and fishing communities, where the oldest animals remained hardest to replace. A species whose life unfolds across centuries can absorb neither rapid extraction nor persistent accidental mortality very easily. Human fishing changed the arithmetic of its survival, and the consequences remain present in every generation that follows. Protection arrived for the Greenland shark only after centuries of extraction had already altered the balance of its population. In September 2022, the Northwest Atlantic Fisheries Organization adopted a measure prohibiting the intentional fishing and retention of Greenland sharks in international waters. For the first time, an international fisheries body had placed this ancient predator under a rule designed specifically to prevent deliberate removal. The change also addressed accidental capture. Greenland sharks, brought up unintentionally, are required to be released rather than retained, although domestic rules in countries with no discard requirements create limited exceptions. Iceland, Norway, the Faroe Islands, and Greenland may operate under those national provisions when a shark is caught accidentally. The distinction matters because international waters are only one part of the animal's range. Coastal and national jurisdictions remain governed by their own regulations, leaving protection uneven across the northern seas. There is something difficult to measure in the date itself. The rule arrived in the same year that the Cornwall shark died, after living for roughly a century without ever being covered by an international protection of this kind. Her lifetime stretched across a period when the species could be harvested for liver oil, caught incidentally in modern fisheries, and largely overlooked by conservation law. The protection, therefore, marks a beginning, not the completion of a long effort. For most species, conservation can be judged within decades. Populations can respond to reduced hunting, protected breeding grounds, or limits on bycatch quickly enough for several human generations to witness the result. The Greenland shark belongs to a different biological calendar. Sexual maturity may not arrive until around 150 years of age, while generation lengths have been estimated at roughly 50 to 75 years. A population damaged over centuries cannot simply rebound because a regulation changes on a particular autumn day. Even complete protection would not make the lost animals return. The older females removed from the population represented reproductive potential accumulated across lifetimes that cannot be compressed into a shorter recovery period. Every young shark entering the population begins a biological journey measured not in seasons, but in generations that themselves span much of recorded human history. That makes accidental capture especially important. An estimated 3,500 Greenland sharks may still be caught as bycatch each year across northern fisheries. Against a population whose true global size remains uncertain, each loss carries a weight that cannot be understood by counting carcasses alone. The problem is not simply how many sharks disappear in one year. It is how many future generations those missing animals would have represented. For an animal with such a long generation time, conservation becomes an act of inheritance rather than immediate recovery. The people who set the rule may never witness its full consequence. A shark protected today could spend another century moving through northern waters before reaching the age at which its descendants become part of the population's future. In that sense, the measure asks humans to think beyond the ordinary horizon of environmental policy, preserving possibilities that belong to lives not yet born. The September decision changed the legal relationship between people and this species. But law moves at the speed of institutions, while Greenland sharks live at the speed of deep time. A protection enacted today can prevent deaths immediately, yet its biological reward may remain invisible for centuries. The first international safeguard therefore carries an unusual kind of promise, one that cannot be measured by the next season, the next decade, or even the lifetime of the people who created it. Its success belongs to a future population that may inherit the sea long after the original decision has become history. The cold itself is part of the Greenland shark's biology. woven into the way its body functions and the places where it can live. Now that boundary is moving. Across the Arctic, temperatures are rising at roughly 4 times the global average, making the region the fastest warming area on Earth. For an animal whose existence is closely tied to cold water, this is not simply a change in scenery. It alters the physical conditions that have surrounded its entire evolutionary history. Sea ice records make that transformation visible from above. Since satellite observations began in the late 1970s, the extent of summer Arctic sea ice has declined substantially, and climate models indicate that ice-free summers could occur within the coming decades under continued warming. As the surface ocean absorbs more heat, the cold water favored by Greenland sharks can retreat downward, narrowing the depth range where suitable temperatures and productive feeding grounds overlap. That shift also changes the geography of opportunity. A deeper band of cold water may still remain available, but the distribution of prey does not necessarily move with it. Seals, fish, and other animals respond to warming through changes in abundance, migration, breeding, and range. A predator that evolved to follow familiar patterns of movement may encounter a landscape in which those patterns are gradually rearranged. The shark does not need to become unable to tolerate warmth for climate change to matter. A change in where prey gathers can alter the energetic cost of finding it. Meanwhile, the retreat of sea ice is opening parts of the Arctic to human activity. Shipping routes are extending into waters that were historically blocked for much of the year. Commercial fishing fleets can operate farther north, while interest in mineral resources and other forms of extraction grows alongside the disappearance of seasonal barriers. The same reduction in ice that changes the shark's habitat can also make that habitat more accessible to the species that share it with us. For a creature with a reproductive cycle measured in centuries, the pace of this transformation is especially difficult. A Greenland shark may require around 150 years to reach sexual maturity. Evolution works through generations, with inherited traits becoming more common when they provide advantages under changing conditions. If the environment changes substantially within a few decades, there may be too little time for natural selection to produce meaningful physiological adaptation before the next generation arrives. That does not mean the species is incapable of change. It means the biological clock is profoundly out of step with the speed of the modern climate. A population cannot manufacture generations faster simply because its surroundings are changing faster. The traits that carried these sharks through earlier climatic shifts were shaped across immense spans of time, not within the duration of a human career. The Greenland shark has already lived through worlds that would be difficult to recognize today. Individuals alive in the distant past hunted beneath seas bordered by glaciers covering large portions of North America and Europe. The species endured repeated advances and retreats of ice, periods of warming and cooling, and transformations in marine ecosystems that unfolded across thousands of years. Those ancient changes were real, but their tempo was different. Geological and climatic forces reshaped the Arctic over timescales long enough for generations to follow one another and for populations to respond gradually. The present transformation is compressed into decades. For an animal whose life is measured in centuries, that difference in speed may be more consequential than the magnitude of the temperature change itself. The Greenland shark has survived changing worlds before, but this time the world is changing faster than its generations can turn. A death that left no descendants became, instead, a source of information. The Cornwall shark entered scientific literature carrying something the species had never provided before, a documented account of what happened when a Greenland shark's body failed. Her postmortem was transformed into a peer-reviewed record, establishing the first verified entry in a category that had effectively been empty, the documented cause of death of this species. That record now gives future researchers a point of comparison. If another Greenland shark strands and its tissues reveal meningitis, the disease can be recognized against an existing case rather than appearing without context. If no later animal shows the same infection, her death will remain an unusual individual event, preserved as the only confirmed example known to science. Either outcome gives meaning to evidence that otherwise would have disappeared with the tide. Her tissues also created a much broader baseline: length, weight, nutritional condition, tissue structure, visible disease, organ changes, and microscopic abnormalities could finally be recorded from a Greenland shark examined after death in the United Kingdom. Before this event, scientists could study living animals through observation, capture, tagging, imaging, or tissue sampling, but they lacked the intimate physical record that only a full post-mortem can provide. The difference is subtle in appearance and enormous in scientific value. Among the most important specimens were the brain tissues and cranial fluids associated with the infection. The bacteria recovered from her were preserved alongside biological material that can be revisited as methods improve. What can be detected in a specimen today is not necessarily everything that future laboratories will be able to detect. New genetic techniques, improved microscopy, and more refined chemical analysis can return to old samples and extract information that earlier investigators could not see. Her eyes carry the same potential. The tissues examined during the post-mortem belonged to an animal whose corneas showed extensive damage, yet whose species has now been shown to retain functional retinas even at extreme ages. Preserved eye material therefore becomes part of a much larger effort to understand how vision survives across centuries of life and how chronic parasitism changes the surface without necessarily destroying the deeper sensory. Machinery. The bacterial isolate may become equally valuable. Pasteurella is widespread, and its presence in a Greenland shark does not by itself explain how infection gained access to the nervous system. Her specimen gives researchers a starting point for examining bacterial characteristics, host defenses, environmental exposure, and the conditions that might allow an ordinarily familiar organism to become lethal inside an animal adapted to one of the planet's most demanding habitats. That is the strange generosity of a specimen like this. Her death cannot be undone, and the missing years of her life cannot be recovered. What remains can still be examined, compared, measured, and questioned by people who may never encounter another Greenland shark on a laboratory table. The preserved tissues extend her scientific life beyond the beach where her biological life ended, allowing future researchers to return to the same evidence with better instruments and different questions. A sample can therefore outlast the circumstances that created it, carrying traces of a disease, a diet, an environment, and a physiological history into laboratories that did not yet exist when the shark was examined. Those traces may reveal relationships invisible to researchers now. For a species that can spend centuries beyond human reach, one accessible body can become an archive. The Cornwall shark provided that archive at a rare moment when nature brought an animal from an almost unreachable world directly into the path of marine science. There is a strange exchange taking place between a very old shark and a very young field of medicine. The Greenland shark has spent centuries maintaining the machinery of its own cells, and now researchers are examining that machinery for clues to problems that human medicine has struggled with for generations. Its value may lie not in making people live forever, but in revealing how damaged cells can remain functional for an extraordinary length of time. Inside the genome are repeated copies of genes involved in repairing damaged deoxyribonucleic acid. Each copy belongs to a larger system that helps detect and correct molecular injuries before those injuries accumulate. Human cells possess repair pathways as well, but the Greenland shark appears to have expanded some of these systems, providing researchers with another natural experiment in how long-lived animals preserve cellular integrity. The significance is not that one shark gene can simply be transferred into a person. The deeper interest lies in understanding which repair mechanisms matter, how they interact, and whether parts of that architecture can eventually guide treatments that slow age-related deterioration. Cancer offers another window. The shark carries unusual modifications involving the TP53 gene, one of the central regulators of damaged cells, along with an expanded nuclear factor kappa B pathway involved in immune regulation and cellular responses. Together, these features may contribute to unusually strong control over cells that have acquired dangerous mutations. Documented cancers in Greenland sharks are extraordinarily rare, although absence of observed disease is not proof that cancer never occurs. Researchers therefore treat the genome as a collection of clues rather than a finished explanation. The same principle reaches into the eyes. A January 2026 study examining Greenland shark retinas found that the light-sensitive photoreceptor cells remained structurally intact in animals more than a century old. That persistence matters because the gradual loss of specialized retinal cells contributes to major forms of human visual impairment, including age-related macular degeneration. Understanding how these cells maintain their structure over such immense spans of time may eventually suggest ways to protect human vision from the cumulative damage of aging. Molecular biologist Paul Robbins has framed the broader goal in a way that keeps the science grounded. The aim is not simply to make people live longer, but to keep people healthier for longer. That distinction changes what longevity means. A century of life is valuable only if the tissues supporting that life can continue functioning. The Greenland shark offers an unusual example because its extreme lifespan appears to be accompanied by mechanisms for preserving cellular stability rather than merely extending the passage of time. Yet the exchange has an uncomfortable edge. Laboratories are studying this species because its biology may contain solutions to human problems involving aging, cancer, cellular repair, and vision. At the same time, human activity has made survival more difficult for the animal carrying those solutions. Fishing removes individuals that may have taken generations to reach maturity. Warming alters the cold environment on which their lives depend. Persistent pollutants accumulate through the food web and remain stored within bodies that may carry them for centuries. The contradiction requires no dramatic conclusion. A species can be both a scientific resource and a living population, deserving protection in its own right. Every surviving Greenland shark carries biological information that cannot be reconstructed from a laboratory model alone. If that information disappears, medicine loses more than a potential source of discoveries. It loses a natural experiment in longevity that began long before modern science existed, and whose full meaning may only become visible after the animal itself is gone. There is a particular kind of knowledge that begins with an absence. In the case of the Cornwall Greenland shark, the missing pieces are not hidden behind a complicated equation or an inaccessible archive. They are simply places where observation has never reached. We do not know how she contracted the Pastorella infection that ultimately became part of the chain of events leading to her death. The evidence confirms the bacterium was present, but presence alone cannot reveal the doorway through which it entered her body. Her accumulated chemical burden adds another uncertainty. Greenland sharks can retain environmental contaminants acquired through their food over immense spans of time, yet the Cornwall examination could not establish whether those substances had weakened her immune defenses before the infection developed. A body can carry a history without revealing precisely how each part of that history affected its final days. The distinction matters because correlation cannot establish causation, especially when the entire known record consists of one animal. Climate introduces another unanswered layer. Warmer Arctic and North Atlantic waters are changing the distribution of organisms, prey, and pathogens. But there is no established record showing whether warming has altered the exposure of deep water Greenland sharks to bacteria associated with shallower environments. The possibility is scientifically important, but possibility is not evidence. For now, the connection remains open. Even the natural ending of a healthy Greenland shark is almost completely outside human observation. A specimen may survive for centuries, yet no scientist has watched one complete its life under normal conditions. We do not know what the body of a 400-year-old shark looks like when age, rather than infection, injury, starvation, or human interference, finally brings its life to an end. There is no established baseline for that final passage. Some of the most basic facts of reproduction remain equally elusive. The gestation period of the species has not been directly observed, and no researcher has watched a Greenland shark give birth in the wild. The timing of reproduction, the precise circumstances surrounding birth, and many details of early life remain concealed within an environment where direct observation is exceptionally difficult. The same limitation surrounds population estimates. The deep Arctic cannot be counted like a coastal colony or a fleet of fishing vessels. Historical catch records can reveal patterns of removal, and those records have been used to reconstruct substantial declines, but reconstruction is not the same as a census. Estimates suggesting losses of roughly 30 to 60% describe what historical evidence allows researchers to infer. They do not provide a precise count of every living shark beneath the northern seas. That uncertainty makes the Cornwall stranding difficult to place. It may have been an isolated medical catastrophe involving one animal with an unusual infection. It may also contain an ecological signal that cannot yet be recognized because there are too few comparable cases. Both possibilities remain consistent with the evidence. What exists, then, is one forensic record from an animal whose life unfolded almost entirely beyond human observation. From that single record, science can establish that something went wrong within her nervous system, that she moved upward from her normal environment, and that she eventually died after entering conditions radically different from those in which her body had evolved. That is not a failure of science. It is the honest boundary of observation. The Greenland shark has spent its existence in a part of the ocean that humans have barely been able to witness, and her death has shown us both what careful examination can reveal and how much remains beyond the reach of our instruments. The history carried by a Greenland shark begins long before the Arctic looked anything like the place we know today. Its broader sleeper shark lineage reaches back tens of millions of years into intervals when northern waters were warmer, shallower, and connected through ocean passages that no longer exist. The animal moving beneath modern Arctic ice belongs to a branch of life that has already experienced worlds almost unrecognizable beside our own. Around 66 million years ago, the end Cretaceous extinction transformed the planet. An asteroid impact disrupted climate, food webs, and ecosystems across land and sea, bringing the age of non-avian dinosaurs to its close. Many large marine predators disappeared during the upheaval. The sleeper shark lineage persisted. Its survival was not a matter of immunity to catastrophe. It was the result of biological traits and ecological flexibility that allowed its ancestors to remain where other lineages vanished. Much later, around 34 million years ago, the transition between the Eocene and Oligocene brought another profound climatic shift. Global temperatures fell, ice expanded, and ocean circulation reorganized. The northern seas entered a different physical regime, and the evolutionary history of sleeper sharks continued within it. Their lineage endured another transformation of the planet without needing to move according to a human timetable, because there was no human timetable yet. Then came the Pleistocene, a long sequence of glacial advances and warmer intervals that repeatedly redrew coastlines, altered currents, and changed the distribution of marine life. Ice sheets expanded across enormous portions of the northern hemisphere, then retreated. Sea levels rose and fell. Entire communities appeared, disappeared, and reorganized around changing temperatures. The ancestors of the Greenland shark persisted through those cycles, carrying forward genetic adaptations shaped by pressures that unfolded over thousands and millions of years. A living animal does not remember an ancient climate in the way a human remembers a place, yet its genome preserves traces of evolutionary history. Every generation that survived difficult conditions passed some combination of useful traits onward, while less suitable variations became less common. Over immense spans of time, selection assembled a biological system capable of functioning in cold water, scarce light, changing prey fields, and an environment that repeatedly shifted around it. That makes the modern Greenland shark a remarkable point of continuity. Somewhere inside its cells are instructions descended through an unbroken chain of survival, extending across geological ages. The same broad lineage that persisted through ancient warming, global cooling, changing oceans, and repeated glaciations now exists beneath waters being transformed on a human timescale. The contrast becomes difficult to ignore when its modern conservation status enters the story. The International Union for Conservation of Nature lists the Greenland shark as vulnerable, reflecting the pressures created by fishing and the species' exceptionally slow life history. Its vulnerability did not arrive with an asteroid or an ice sheet. It emerged from contact with a much younger force. Homo sapiens has existed for roughly 300,000 years, while industrial-scale fishing has occupied only a small fraction of that history. Yet within that brief interval, humans have become capable of removing animals from the ocean faster than a species that takes generations to mature can replace them, while also changing the temperature and chemistry of the habitat around them. The Greenland shark survived planetary upheavals that lasted for ages. Its greatest uncertainty now comes from a species whose entire industrial history is barely a moment beside the time written into the shark's lineage. The difference is almost geological. A lineage shaped across deep time is now being tested by changes compressed into a single human lifetime. There is a strange arithmetic hidden inside the life of a Greenland shark, because the numbers do not behave like the numbers of most animals we know. The Cornwall shark had lived for roughly 100 years, yet she had never reached the age at which females are believed to reproduce. A century of survival had accumulated inside one body, but none of that survival had yet become a new generation. For a species with such a long beginning, reproduction is an unusually distant event. A female may not reach sexual maturity until around 150 years of age. If she eventually lives to 400 years, her possible reproductive span could extend across roughly 250 years. That is not a promise of offspring, only a biological opportunity dependent upon survival, suitable conditions, adequate prey, and the absence of disease or accidental capture. Before that window even opens, the animal has already spent a century and a half growing through an existence that demands extraordinary patience from evolution. Every mature female, therefore, represents an investment measured in generations rather than seasons. By the time reproduction begins, her body has carried the accumulated result of countless successful years. She has survived changes in prey, predators, temperature, currents, and the hazards created by an increasingly crowded ocean. When one such animal disappears before reproducing, the loss is not simply the removal of one individual from a population. It is the removal of a biological future that required more than a century to arrive at the threshold of possibility. Against that timescale sits another number. Estimates suggest that roughly 3,500 Greenland sharks are accidentally captured and killed each year through bycatch. The total global population remains uncertain, so there is no precise denominator against which to measure those losses. What can be understood is the mismatch between removal and replacement. A population whose members may require 150 years to mature cannot compensate rapidly when mature or developing animals are removed faster than generations can turn over. The Cornwall shark makes that arithmetic especially tangible because her life ended before her first opportunity to reproduce. Her death did not erase a known lineage of descendants. There were none. What disappeared was the possibility that the century she had already survived might eventually have become part of the species' future. Even the existence of her scientific record depended on a chain of ordinary circumstances that had nothing to do with the shark herself. A dog walker happened to cross that stretch of Cornish beach on that particular morning in March. Researchers happened to be able to reach the carcass before decomposition erased much of what could be learned. The body was recovered, examined, and preserved sufficiently for science to extract information from an animal that had spent its entire life beyond ordinary human observation. A slightly different tide could have carried her farther from shore. A different week could have left the beach without the nearby vessel traffic that helped bring attention to the stranding. Decomposition could have reduced the remains beyond meaningful examination. In that case, her body would have returned to the same ocean that had sustained her, and the species would have remained without a documented forensic death record. That is the disturbing part of the arithmetic. For most of her century, nothing about her existence was visible to us. Then, for a brief interval, circumstance placed her within reach of human science. One life became measurable, one death became evidence, and one vanished possibility became part of the record. A beach in Cornwall belongs to a very different chapter of the ocean's history. Along this coast, human settlement reaches back beyond the Norman conquest, and the waters around Penzance have carried fishing boats and trading vessels for more than 1,000 years. Villages, harbors, stone walls, and familiar paths have grown around a shoreline shaped by generations of people. Yet in March, one of the oldest animals alive on Earth arrived there from a world that had almost nothing in common with it. The Greenland shark had never evolved for this coastline. Its ancestors had spent millions of years in cold northern seas, under conditions where temperature, pressure, darkness, and seasonal change followed patterns entirely unlike those of southwestern England. There was no evolutionary history preparing this animal for shallow Cornish water, for kelp moving beneath a brighter surface, or for the irregular contours of a coast inhabited by humans for centuries. Her arrival was not a journey she had chosen. Something had disrupted the biological systems that normally allowed a deep ocean animal to orient itself and remain within the environment suited to its body. The bacterium associated with her meningitis had reached her brain, and with that infection came the possibility that navigation itself had become unreliable. The ocean then carried her beyond the boundaries that had defined her existence. That connection between distant seas is easy to overlook from land. Water does not recognize the borders drawn on maps. Currents move heat, nutrients, organisms, and microscopic life between regions separated by thousands of kilometers, about thousands of miles. The Arctic Ocean is not an isolated chamber. It belongs to a planetary circulation system in which water can eventually pass through distant basins, carrying traces of one environment toward another. For the person walking along that Cornish beach, none of this would have been visible in the morning. The scientific record does not preserve the identity of the dog walker who discovered her. That person was simply moving through an ordinary part of an ordinary day, following a familiar route beside the sea. Yet that routine moment intersected with something that had never been documented in British science before. For approximately 2 days, the shark remained somewhere between the coast and the harbor, moving through water that bore little resemblance to the deep habitat in which her body had been shaped. She was no longer following the conditions that had governed her life for a century. The change was not merely geographical. Temperature, pressure, illumination, sound, currents, and the distribution of familiar prey would all have differed from the environment in which her senses and behavior had developed. Then there was the light. For an animal whose normal existence unfolded far below the surface, daylight would have represented an entirely different sensory world. As she moved into shallower water, increasing amounts of sunlight could penetrate the sea around her. The retinal cells that had remained structurally intact through more than a century of darkness were suddenly receiving a stimulus far removed from the conditions under which they had spent their lives. And so the final location of her life carried an almost impossible contrast. A Greenland shark, shaped by ancient cold seas and adapted to the depths, came to rest beside one of Britain's most familiar coastlines. Behind her lay a lifetime no human had witnessed. Around her were beaches, harbor waters, and people following routines that had continued for generations. She died in daylight, at the edge of a world her species had never been shaped to inhabit. There are animals that survive by mastering their surroundings, and there are animals that survive by asking almost nothing from them. The Greenland shark belongs to the second kind. Its lineage has persisted through immense changes in the planet, across eras when oceans warmed, cooled, emptied of oxygen, and filled again with new forms of life. Long before the species itself appeared, its ancestors were already moving through a changing northern world. Mass extinctions came and went. The end of the Cretaceous erased the dinosaurs and transformed marine ecosystems. Other upheavals altered ocean chemistry, currents, temperatures, and coastlines. Glacial cycles repeatedly rearranged the Arctic, while periods of warmth opened waters that would later become covered by ice. Through these changes, the sleeper shark lineage continued, not because it could outrun catastrophe, but because its biology demanded so little from each moment of existence. Its metabolism is remarkably restrained, growth is slow, movement is economical, energy is conserved rather than spent. A body that can survive in cold water does not need to maintain the rapid biochemical pace common among many active predators. The result is an existence built around conservation, where every calorie matters and every unnecessary expenditure becomes something evolution has gradually removed. That strategy has consequences measured on an almost geological scale. A Greenland shark can move through the water at less than walking pace, yet continue doing so for years, decades, and potentially centuries. It does not need to dominate its surroundings. It occupies them, drifting through a realm where scarcity itself has become part of the design. Its success is not written in conquest, but in the extraordinary ability to remain present while the world around it keeps changing. It waits for opportunity, consumes what can be found, and allows time to perform much of the work that speed accomplishes in other animals. The same pattern reaches into reproduction. Sexual maturity arrives extraordinarily late, meaning that generations turn over at a pace almost impossible to reconcile with human experience. Evolution has shaped a creature for persistence rather than rapid replacement. When conditions remain favorable, that strategy can be remarkably successful. When conditions change suddenly, the same slowness becomes a vulnerability. Nothing in that ancient biological history prepared the shark specifically for humans. The behavioral patterns carried by its nervous system were established long before our species became a force in the ocean. Its immune architecture was shaped without fishing fleets, synthetic chemicals, engines, plastic waste, or warming caused by industrial emissions. Even the chemistry of its tissues evolved in a world where there was no human technology capable of altering an entire ocean. That absence matters because evolution does not prepare organisms for every possible future. It responds to pressures that have already existed. A lineage develops defenses when repeated generations encounter a threat, and individuals carrying useful traits leave more descendants. No such process could have produced a Greenland shark adapted to long lines, industrial pollutants, or rapidly changing Arctic temperatures before those pressures existed. For hundreds of millions of years, the great lesson written into this lineage was persistence. Cold arrived, warmth returned, seas changed, predators disappeared, and new ecosystems formed. The shark endured by remaining within the narrow boundaries its body could sustain, conserving energy and allowing time to become an ally. Then humanity entered the ocean with tools unlike anything that had shaped the species before. There was no inherited behavior for this encounter, no ancient defense hidden within its genome, no evolutionary memory capable of recognizing a fishing line or a warming sea. The animal that had survived almost everything had simply never needed to survive us. Somewhere beneath the Arctic ice, in water close to freezing and nearly 2.5 kilometers, about 1.5 miles, below the surface, Greenland sharks are still moving through a world almost entirely beyond human observation. Their pace can fall below 1 kilometer per hour, about zero point 0.6 miles per hour, as they travel through immense spaces where pressure, cold, and darkness have remained familiar companions for generations. Some of those sharks were born when the first airplane took to the sky. Others were already alive before the American Civil War had begun. A few may have entered the world before European maps had given the Arctic coastline anything resembling its modern outline. Their ages turn ordinary human history into a brief interval. A harbor changes, a nation changes, a technology appears, spreads, and becomes obsolete. Far below, a shark can continue through all of it. Above them, however, the physical world is becoming different. The Arctic is warming rapidly, and the frozen surface that once formed a broad seasonal ceiling is retreating. Less sea ice leaves more open water exposed to sunlight, weather, ships, fishing activity, and resource exploration. The change does not arrive as a single event. It alters temperature gradients, currents, prey distributions, and the seasonal boundaries between one habitat and another. The sharks beneath that changing surface cannot recognize the cause. They cannot know that the ice above them is thinning, or that routes once blocked for much of the year are becoming accessible to vessels. Their biology responds to temperature, pressure, chemistry, and food availability, not to the human meaning attached to those changes. There is another disturbance moving downward through the same water. Each year, thousands of Greenland sharks are accidentally captured in fishing gear intended for other species. The figure of roughly 3,500 deaths represents an annual removal whose significance becomes difficult to grasp beside a reproductive cycle that can stretch for roughly 150 years before maturity. A shark brought upward through hundreds or thousands of meters of pressure change may survive the capture, or it may not. For many individuals, the outcome is never observed. None of these animals knows Cornwall. They do not know about the young shark, roughly 4 meters, about 13 feet long, who emerged into shallower water after a lifetime shaped by the deep. They do not know that humans recovered her body over 2 days, examined her tissues, studied her brain, preserved her eyes, and turned one unexpected death into a permanent scientific record. That record now sits beside a much larger reality. The ocean she left behind is still there, and Greenland sharks are still moving through it, carrying bodies shaped by ancient conditions into a century of unprecedented environmental change. Their lives continue according to rhythms that began long before modern civilization. Around them, those conditions are shifting on a human timescale. For a species whose generations unfold across centuries, that difference matters. A landscape can change within a lifetime. A population can decline within decades. Evolutionary adaptation requires generations, and generations for this animal arrive slowly. The deep Arctic remains vast, but vastness is not the same as permanence. Even a habitat that appears limitless can become fragmented when temperature boundaries shift and prey follows them into unfamiliar regions. For an animal adapted to persistence, the speed of environmental change may matter as much as its final destination. Somewhere beneath the ice, another Greenland shark is moving through the cold, unaware of the history unfolding above. The water still surrounds her as it surrounded generations before her. Yet the world, carried by that water, is no longer quite the same. For a century, the Cornwall shark moved through an existence that humans could barely observe. Then, on an ordinary stretch of coast, her body became the first Greenland shark body in marine science to yield a complete account of its own death. The discovery did not reveal a spectacular struggle or the attack of another predator. It revealed something much more familiar, a bacterial infection caused by Pasteurella, a bacterium capable of reaching the brain and disrupting the systems that allowed her to navigate. Inside her tissues, the evidence described an illness that had progressed beyond the point where her remarkable adaptations could compensate. Inflammation had spread through the brain, and fluid had accumulated where healthy neural tissue should have remained functional. The animal that had spent roughly a century moving through extreme cold had reached a place where direction itself could no longer be trusted. Her body had endured an environment that would challenge most vertebrates, yet an infection involving an organism found elsewhere in nature had become fatal. There is a strange precision in that contrast. The Greenland shark carries biological systems shaped by immense evolutionary time. Its cells possess mechanisms involved in repairing damage, maintaining proteins, and coping with the demands of an unusually long life. Those systems are now being studied because they may reveal principles relevant to human aging and disease. Yet none of that tells us exactly why this particular infection overcame this particular animal. The genome can preserve extraordinary capabilities without making an organism invulnerable. That uncertainty is what remains after the measurements have been taken and the tissues have been examined. Science can identify the bacterium. It can document inflammation, neurological damage, and the physical changes associated with disease. It can preserve samples and compare them with future cases. What it cannot yet provide is a complete chain of events connecting the first moment of infection to the final loss of function. Her body has therefore become part of a record, much larger than the beach where she was found. Tissue samples remain within scientific collections, including material held by the Zoological Society of London. The findings have entered peer reviewed literature, giving future researchers a reference point that did not exist before her death. If another Greenland shark is eventually examined after stranding, its tissues can be placed beside hers, and differences that are invisible today may become meaningful through comparison. For now, though, her case remains singular. There are no thousands of carefully documented deaths against which her illness can be measured. There is one complete forensic record, surrounded by generations of animals that disappeared beneath the sea without leaving evidence behind. The empty space around her record is part of the science itself. Somewhere beneath the Arctic ice, another Greenland shark is moving through cold water in a darkness it has known since birth. Its heart may beat only about 5 times each minute. It has no reason to recognize the world above, and no way to know that its species has become an object of intense scientific attention. It may live for centuries, carrying biological information that began accumulating long before human civilization. The Cornwall shark gave science something rare, a death that could be examined closely enough to become knowledge. She also left behind something less tangible, a boundary around what remains unknown. Her body answered some questions, opened others, and showed how little of this ancient life we have actually witnessed. The ocean continues beyond the reach of our records, and within it, her kind continues moving through a world whose future now depends partly on choices made far above the water.